part of this 3-part interview series where we'll be looking at bioanalysis for biosimilars. We're going to jump straight back in. So, Stacey, a key best practice in PK similarity studies seems to be using a single method to measure both the biosimilar and the reference product. What are the main advantages of that approach compared to using, say, separate methods, particularly in terms of accuracy, consistency, and regulatory compliance?
There isn't specific regulatory guidance on how to perform the PK bioanalytical methods for biosimilar programs. So we follow the ICH-M10 guidelines on bioanalytical methods for validation. Previously, we would follow the earlier FDA and EMA bioanalytical method guidance. A very helpful resource on this is coming from the 2014 white paper from the AEPS ligand binding assay bioanalytical focus group. This provides a systematic framework of demonstrating the bioanalytical similarity between the biosimilar and the reference product. The paper strongly recommends developing a single ligand binding assay. Once you've established that a single assay performs equivalently for both the biosimilar and the reference product, there's some significant advantages. You have increased accuracy and you've reduced your bias. So using one method minimizes systematic differences that could arise between two methods. It gives greater confidence that any observed difference or not difference is coming from the molecule itself and not due to the assay. At that point, you'll have the consistency, you'll have lower variability. So looking at a single assay, you'll take away any of the assay bias. So any between assay variability that you may have using two methods. Also, all of the study samples, whether from the biosimilar or the reference product, will be analyzed within one method rather than analyzing both samples into separate methods. There's also benefits from an operational standpoint. You can analyze all the samples together. You're going to reduce your analysis time, your cost, and the logistic complexity. From a regulatory perspective, you're going to have a cleaner data package. So you'll have one set of data for all of the samples that are coming from a single method, rather than having two sets of data and needing to do an additional statistical comparison to determine if there is a difference and if that is potentially related to assay. However, this will be everything's in one package under one method. So the one assay approach is widely viewed as the best practice because of these reasons. So they're reliable, the comparable data, ultimately strengthening your overall biosimilar package and showing the similarity between the biosimilar and the reference product.
In method development for these PK assays, what specific tests or maybe parameters need extra evaluation or extra validation potentially when dealing with biosimilars, and how does this differ from the requirements for, say, regular molecules or non-comparative studies?
For the standard PK method used in new biologic or non-comparative studies, we follow the usual method development steps. We're going to optimize the reagents, check sensitivity, selectivity, matrix effect, and anything additional that may go into developing a single method for a non-comparative study. When supporting a biosimilar study, the main goal is a single assay and to show the comparability between the biosimilar and the reference product. The most important step during method development is going to be a head-to-head comparison of the biosimilar and the reference product. We'll prepare calibration standards in using both molecules. We'll run these on the same plate, preferably over at least 3 runs, additional analysts, over multiple days. When we statistically assess the similarity, we want to see if these curves are going to overlay. So we're going to visually see if there's any meaningful differences, as well as looking at the data points and to see if there's any statistical difference. between the curves. We're going to use a similar approach that we would evaluate for any PK method. Are they within 20% of each other? Once we've determined that the calibration curves are similar, then we're going to look at the QCs. We're going to prepare the QCs in different with both molecule, and that'll be further assessed during the method validation.
So during that method development then, for biosimilar PK assays, what unique considerations or additional tests are critical for biosimilar PK assays specifically?
In addition to the standard validation parameters that are outlined in the ICH-M10, the biosimilar PK validation requires specific evaluation of QC samples prepared from both the biosimilar and the reference product. This includes assessments for selectivity, accuracy, precision, short and long-term stability, and dilution linearity may also be evaluated in both the biosimilar and the reference product. This dual evaluation is going to add complexity to the validation, as well as the volume of the validation work. But it is essential to demonstrate the similarity between the biosimilar and the reference product in order to support the use of a single assay. This is going to ensure the robust results, so the validation needs to be strategically planned. We'll prepare QCs in both the biosimilar and the reference material. We're going to ensure that both sets of these QCs are on the same plate. For the calibration curve, we'll use the biosimilar, and then we'll set this up to show comparability within the QCs across different analysts, across different days, and across different runs. So one of the common pitfalls that we do see is just strictly evaluating the QC data for bias. So if we are seeing acceptable data within the QCs, we're going to need to take that one step further for the biosimilar program. For example, if the biosimilar QC recovers with a +15 bias and the reference product recovers with a -15 bias, within themselves, they are going to meet the acceptance criteria. But for the biosimilar program, we're going to take that a step further, and we're going to look at the difference between the two. So they're going to show a 30% difference, which is we're not going to be acceptable in that. So that's one of the criterias that we're going to need to look at for the biosimilar program is the difference in the QCs compared to each other. In order to avoid this, we want to make sure that we have a really strong method validation plan that outlines this acceptance criteria to show what the difference is between that biosimilar and the reference QC as well. We're also going to evaluate selectivity within both the biosimilar and the reference material, as well as the short and long-term stability. We'll prepare those both with both molecules. And so during the validation, we're going to ensure that we're rigorously testing both molecules and making sure that we're meeting acceptance criteria to support the use of a single assay.
I was wondering if you'd observed any trends or challenges in conducting PK studies for biosimilars, and are developers adapting to those evolving regulatory expectations in this area?
The regulatory expectations are evolving. Both the FDA and EMA have de-emphasized the need for large comparative phase 3 efficacy studies, placing greater weight on the robustness of the bioanalytical method for both PK and ADA. This demands a higher level of due diligence in the bioanalytical laboratories during method development and method validation, as well as in study sample analysis. Another emerging trend is the increase in single-kit analysis instead of the traditional duplicate analysis. This is also going to increase the method validation work to show that there is no difference using a single-kit analysis rather than a duplicate. This will improve throughput and reduce the cost. However, this is adding, an extra layer, especially with validating the biosimilar as well. So labs must be careful making sure that the single method for a biosimilar is robust. It shows the comparability, as well as if they're going to introduce the single-kit analysis.
How do regulatory agencies like the FDA, the EMA, how do they evaluate PK similarity data? And are there criteria or thresholds maybe that developers need to meet to demonstrate that comparability?
From a bioanalytical standpoint, the FDA and EMA are placing a significant weight on the quality and the reliability of the PK assay when evaluating the similarity data. The agencies are reviewing PK data sets, not just for the clinical pharmacology outcomes, but also evidence that the underlying method itself is demonstrating bioanalytical similarity between the biosimilar and the reference product. This approach is going to help minimize method-related bias and support the demonstration of bioanalytical similarity. This is a significant increase in the complexity of the validation, with key challenges including need to evaluate the dual QCs from both the molecule to apply tighter acceptance criteria. A major expectation is the implementation of the single assay, which we've talked about with the validation. And so just adding to some of the criteria that I did previously talk about with the validation, running the dual sets of QCs for both the QC and the biosimilar across all validation runs, as well as looking at some of the other tests, specifically your selectivity. Also establishing and meeting tighter criteria, specifically when you're looking at the difference between the two sets of QCs. So rather than just evaluating whether they're in acceptance of themselves based on their nominal concentration, also looking at the percent difference that they are from each other. And so the recent FDA and EMA guidelines reflect greater confidence at well-characterized analytical and PK packages and reduce the clinical requirements and making sure that the bioanalytical data quality is high. Developers are adapting by building more rigorous validation plans, conducting early regulatory consulting on assay strategies, and carefully justifying any deviations or any non-traditional duplicate analysis, and making sure that these are discussed, and if they do need to get additional follow-up from any of these agencies, that they're reaching out early and on a regular basis.
Great, thank you so much, Stacey. So we're going to wrap it up there for part 2, but please join us for part 3, where we're going to be looking at immunogenicity assessment for biosimilars.